morning, students. We are studying right now bacteria. In our last class, we studied about the genetic material of bacteria, and we studied also about plasmid DNA, the extra chromosomal material, circular material in the cytoplasm are called plasmids, and there are various forms of plasmids we discussed in our last class. Today we will discuss about cytoplasmic organelle in detail. What are other cytoplasmic organelle present here? So the first topic is cytoplasmic organelle. Or content. So the prominent structure inside bacterial cell is mesosome. Mesosome is a structure which bacterial cell has formed by inward valination of the cell membrane. Such invalination inward develops rooted vesicles and projections in the cytoplasm, and they store several types of enzymes required for energy production and other metabolic processes is present here in this structure. So the structure mesosome is also associated with energy production. Apart from energy production, mesosome also helps the cell in the process of cell division, which is common, is binary fission in bacteria. So the inward great invalination of cell membrane which stores several type of enzymes for energy production and other biomolecule synthesis. So mesosome also play a role in binary fission. How? First, the mesosome starts division and creates a furrow in the cell, due to which the cell divides into two halves. At the same time, a copy of genetic material passes into another half. And in this way, the furrow becomes deeper and deeper with time, and a time came when the cell divides into two equal halves completely. So this is mesosome, a cytoplasmic structures. Cytoplasmic content will be more appropriate with bacteria. So mesosome is one cytoplasmic content. Third thing about mesosome is generally in bacteria, genetic material is close to the mesosome. genetic material is embedded closely to the mesosome. Site of energy production. Site of energy production. Helps in biology. So this is all about mesosome. We come upon another cytoplasmic content inside bacteria is ribosomes. Bacterial ribosome. It is of 70S type. 70S type. There is a smaller rRNA and bigger rRNA. Two parts are in 70S type of ribosome. The first part is, which is smaller in size, is 30S and the bigger one is 40S. Combined they are 70S. S here is Schwedt per unit. S represents Schwedt per unit. What is Schwedt per? Schwedt per is equivalent to 1 into 10 to the power minus 3 13 seconds. In this time, the movement of ribosome in cerium chloride gradients is taken as the measurement of their heaviness. So, if in this time a ribosome travels 70 micrometer, then it is called 70S. If it travels 80 micrometer, then it is called 80S type of ribosome. So, this is a mechanism on which we calibrate, in which we differentiate among different sizes of ribosomes, as ribosomes are granular structure in the cytoplasm. So, cytoplasmic ribosomes are of 70S type, and sometimes they are associated with one another in the cytoplasm. Such a structure is called polyribosome. Polyribosome. Ribosomes are protein factory inside cytoplasm. Ribosomes are protein factory inside cytoplasm. Apart from ribosomes, there is one more structure in photosynthetic forms of bacteria, known as chlorosome. Sometimes the cell membrane move invaginate inside and form a vesicle-like structure, and if that vesicle-like structure stores photosynthetic pigment. They are known as chlorosome. Apart from chlorosome, some lamellae filamentous structures stores photosynthetic pigment like bacterial chlorophyll and known as lamellae, photosynthetic lamellae. So, photosynthetic forms have photosynthetic forms. Have lamellae in cyanobacteria, chlorosome in sulfur bacteria, so which are non-photosynthetic will not have such structures in them. Now become our reserve food material. Reserve food in bacterial cytoplasm. Reserve food is in two forms: organic and inorganic. In organic forms, we have glycogen as reserve food in bacteria. Glycogen, oil droplets, PBH, polyhydroxybutyrate, PHP. These are the organic components present as reserve food in bacteria. Inorganic, volatile granules, are inorganic dyes present in different bacteria and give different coloration. Is volatile granules. Apart from this, the bacterial cell have different inorganic compounds such as calcium, silicate in their cell wall, or many other inorganic constituents may present in the cytoplasm inside a protein covering. So, these are the reserve foods in bacteria. Now, we move towards processes. What are the processes? Life processes. And how does it occur in bacteria? We start with nutrition. Bacteria exhibit both mode of nutrition, autotrophic as well as heterotrophic. Autotrophic as well as heterotrophic mode of nutrition. Among autotrophics, we have two types of photoautotrophics and chemoautotrophics. Photoautotropics have four groups. For example, green sulfur bacteria, green non-sulfur bacteria, then we have purple sulfur bacteria, and purple non-sulfur bacteria. Sulfur bacteria. We can divide this group in two forms. One will be anoxygenic and the other will be oxygenic photosynthesis. Now, in oxygenic section, we have only one eubacteria which is called cyanobacteria, which perform oxygenic photosynthesis, whereas all other forms, these are anoxygenic, they perform anoxygenic photosynthesis. Anoxygenic. How? We can say, whereas in chemoautotropic bacteria, the energy required for converting the inorganic constituents into organic constituents derives from chemical reaction. That's why this form of nutrition is called chemoautotropic nutrition. And the bacterial species which perform this mode of nutrition are Nitrosomonas, Nitrobacter, Pseudomonas. We have many other forms. So, what happens in cyanobacterial photosynthesis? Cyanobacterial photosynthesis is C6H12O6 plus 6O2, which gives 6CO2 plus 6H2O. Reverse, this is respiration. What is its reverse? 6CO2 plus 6H2 in presence of sunlight and chlorophyll convert into C6H12O6 plus 6O2. That means the form of photosynthesis in which the inorganic constituent converts into organic constituents in presence of sunlight and chlorophyll, and oxygen evolves there. So this form of photosynthesis is anoxygenic. But in some bacteria, the water is not present. In presence of oxygen in water, they may have hydrogen sulfide. Hydrogen sulfide, 6 molecule here leads into C6H12O6 plus carbon S sulfur, how many? 6S. Molecule of sulfur will be there. So, here oxygen does not evolve. And the source of the electron acceptor is sulfur, then such bacteria is called sulfur bacteria. If it is green in color due to chemical composition, it is green sulfur bacteria. If it is purple in color, then it is purple sulfur bacteria. If they utilize source other than sulfur, then they are called non-sulfur bacteria. If non-sulfur bacteria is green in color, it is non-sulfur green bacteria, green non-sulfur bacteria. And if it is purple bacteria and does not utilize the sources related with sulfur, it is called. So in that case, what happens? In that case, we have a carbon source that may be CO2 and may be other, and there may be any source of hydrogen other than sulfur, then we will form C6H12O6. In that condition, we must have 24H plus 6, 12 plus 6 H2 और ना 12 So in this form also there will be no evolution of oxygen. So we can see that this type of reaction will be performed in case of green non-sulfur bacteria and purple non-sulfur bacteria. The rest of the sulfur bacteria will perform that will have any source of sulfur with which they will produce organic compound. So the differences in oxygenic and anoxygenic is clear. The reducing agent in oxygenic form of photosynthesis is NADPH2 as reducing agent, whereas in anoxygenic we have NADH2 as reducing agents. In bacteria, oxygen evolves in oxygenic form. Oxygen does not evolve in anoxygenic form. The electron donor is water molecule in oxygenic, and in anoxygenic it is other than water molecule. That may be sulfur source and non-sulfur source. So this is all about autotrophic mode of nutrition. We have left only chemoautotrophs. Who are chemoautotrophs? So chemoautotrophs obtain energy for chemical reaction. For example, A breaks into B and provides more energy. Then this chemical energy will be utilized to reduce any carbon source. For example, it is hydrogen molecule here. That will be utilized for converting inorganic constituents into organic constituents, C6H12O6. So, if energy used in forming the organic food from inorganic constituents is from a chemical reaction, then it is called chemoautotrophic mode. And the forms which do it are several, such as nitrosomonas, nitrobacter. We have pseudomonas. We have iron bacteria. So they all perform chemoautotrophic mode. Now we come over heterotrophic mode. In heterotrophic, we have three types of bacteria. One is called decomposer. The other may be parasites. And some parasite may be symbiotic. So Decomposers are present in detritus matter and are involved in the process of mineralization, putrefaction, and other mineralization processes, formation of humus, breakdown of organic molecule into inorganic constituents. So, decomposers are many in the nature. In the same way, we have parasitic form. They may be ectoparasite or endoparasite. And they may be symbiotic too. For example, Escherichia coli present in large intestine is symbiotic. But when it infects the urinary system, it is called parasitic. So one who is symbiotic may be parasitic at another place. So in parasitic forms we have Mycobacterium tuberculei, we have Bacterium vibrii, we have Helicobacter pylori. So these are parasitic forms, endoparasites. And symbiotic form may be those who are in symbiotic association with another. They do not harm one another. They are necessary for survival of that life form. That structure is necessary. For example, lichens, symbiotic association. Then we have symbiotic association of bacteria with leguminous plant, such as rhizobium. Non-leguminous association we have in Frankia. We have several forms of bacteria present in soil, which is associated with nitrogen fixation in soil. So, heterotrophic forms of bacteria are also present in nature, and autotrophic forms are also present in nature. So this is all about nutrition in bacteria. In next session, we will study reproduction in bacteria. Till then, thank you. If you have not understood anything, please contact me and get your doubts clear early. Till then, stay at home, stay safe. Thank you. Bye.